yeah, this is a different kind of environment than we got the other rock sample for the bio study. These pillows look like a good comparison. So Jess, when you feel like you're in a good spot and seeing these rounded rocks that look like of a size we could pick up, let me know.
Sure thing. Yeah, sure thing. I'll kind of swing it out a little bit since Argus will swing in it, and then I'm sure this rock type will hopefully continue up there as well.
Yep. Yeah, we'll see what we get.
Roger.
A little small coral there.
Looks like a hemicorallium precious coral.
Oh, and there's a long whip of some variety ahead of us. Two of them, actually. Jess, can you pick one of those long stock features at the top of the screen to look at?
Yeah, sure thing. Sure thing. Then we're in a good spot over here as well to pick up the samples.
Okay.
If you see anything that looks resemblant of what you wanted before.
Right. That's going to be pretty long.
Roger.
Can we get a closer zoom on that when it makes sense?
Yeah, sure thing. Go ahead, Jeff.
All right. Our friend, the bamboo coral, is that what we're looking at?
That's it.
All right. Nice long one. Thanks, Jess and Jeff.
Yep, sure thing.
Okay. Actually, before you pull full back, can I look at the rocks a little bit?
We can set down over there, and then you probably get a better view.
Yeah, that sounds good.
Okay. Full way there, please. It was to the right of this one, wasn't it? The rocks you're looking at.
Yeah, I was just trying to get an idea of, they look a little bit less like pillows, more like sheet flow here, right in this feature to our right. That's probably not what I want right there.
I see. You want to go back to the
but
left?
Maybe over to the left. Right.
Roger.
Looking for more of like the rounded features. For those of you tuning in from Hawaii, feel free to
Drop a note in the chat. Where are you? How'd you hear about us? We are in the neighborhood.
With that said, if you guys are just joining us and stuff like that, both OET and Papahanaumokuakea, the affiliated Hui or groups and stuff like that, have a lot of cool educational resources and a lot of the history about some of the previous finds. So if you're just as interested about what we're looking at now and what we're looking to maybe discover or learn, there's definitely a plethora of information online. So for instance, I'm looking at one of the resources, one of the newer resources that just came out for the monument, and it's called it's a deep sea. What is this? Oh, sorry, a deep water resources of the Northwest Hawaiian Islands, and so it sort of covers everything from new biological species that are covered in the area, some of the technologies that we're using. It's a pretty cool blend of not only culture but also maritime history in this area. I think that also on the OET YouTube website, there's a lot of cool information about like highlights of previous expeditions from other stuff like that. And so, and I don't think it would be too far-fetched to say that some of the highlights that we maybe see in this expedition might make their way to the YouTube channel later or some other highlights. So please be on the lookout for not only what's going on during the expedition and before, but also some of the products or some of the research that will be coming out after this. Yeah.
Great. So Beth, is anywhere in the center view here? Oh.
Yeah, so kind of these like little lumpy things. Maybe some of these are down below us.
All right.
Challenge is going to be if any of these are actually loose enough to pick up.
Yeah, look intact. Let's have a look.
Yeah, so maybe, yeah, if we're looking at this on the ground, where are lasers? So maybe like these pieces might be suitable.
Okay. We can go in there and give a poke.
Do we think that's an anemone off to the right there?
Looks like one.
Well, it's a small coral. And a little shrimp.
What about the ones on your screen, the nodules?
I'm sorry, could someone say that again?
Oh, the rock in the center screen for the nodules?
Yeah, these.
Oh, the ones up there. All right. Hey, Jake, want to get out the arm? You see the ones that she was pointing at?
Yep.
Cool.
Am I going to be able to reach that?
Probably not.
Are we going to try that with the port manipulator? Or even the starboard one getting it from so far?
Yeah. Sometimes it's deceiving how far out it can go.
Yeah, so if we want to go for that one right there, see if we can get it.
Steve, where are we going to go with the sample here?
It'll go in the front bio box port side.
Port side, Raj.
All right, so which one of these were I thinking? Yeah, look.
That's the one.
Yep.
That's the money.
Might not be
Be a little
ready
bigger.
to leave this mountain, huh?
It's a pretty big one.
That's not moving.
Okay. What about this?
Yeah, all right.
Okay.
Jeff, gonna push in there a little, please? That's great. Nice. It's also consolidated. Oh, nice.
Yeah. Got it.
There you go.
Yay.
Nice job.
Dentistry school is in
Session.
session.
Oh, that looks really nice and altered.
DVL
Awesome.
reset.
Sure thing. Jeff, you gonna push on in? Nice.
It's got some nice crust as well as some really, looks like some clay secondary alteration of the original basalt. So that's good.
Awesome. Looks
Thank
good.
you.
Right.
Forward. So the only thing about this guy is that since we're putting it in the forward, those coral cutters are going to open like uber amount, and the H camera's right here.
All right, so I gotta be careful about that. Okay.
Yeah, so maybe I angle it down so that it's going to open more into the bin.
Okay.
You know what I mean? And keep your finger on the trigger so that it's good.
So for those at home,
Are you back to?
Yeah, we're full rack back right now.
one of the fun parts of these dives is the training that happens during them. Always quick watch.
Show the folks at home.
What's that?
Show the folks at home.
Oh, yeah.
Oh yeah, there we go.
To see who's controlling the arm right now.
This is really just my attempt to keep the cameras on other people instead of just on us in the back row.
Glad we don't have one of those cameras because it's not as tidy back here.
That's great right there, actually, Jake.
A lot of scientific papers shuffling around.
That's right. Lots of science happening.
Nice.
So
Nice. Well done.
Beautiful.
Yay.
Yay, Jake.
Yay.
Lovely.
is that 51?
That's right.
Great.
All good.
51. Got it.
And I'd like to take a water sample here as well. But maybe wait just a moment for some of this debris that we just kicked up to clear.
Sure thing. All right, Jake, you want to get back
Oh, yeah.
up? For these, since it's a snaggle tooth, if you can keep the jaws closed and then just rotate it.
Yep.
And I'll get you on bubble here.
Is it? Was the last nest 51? This is 52. Oh, never mind.
No, the rock is 51, so 52 is the skin.
Okay.
Yeah.
All right, you got the view. Can watch.
All right, which one am I going for?
Try 4. That's a bit easier for these jaws.
Is that the orange one?
Orange one. The green one's kind of hidden in there.
Yeah.
Negative. Probably should fix that.
So if you have your quad view on, you can kind of sync this up here as the manipulator arm pulls the orange trigger and then look for the tops, the pop on the top.
Got eyes on the camera here. Nice.
Oh,
There you go.
done.
Very nice.
Yay.
Awesome.
All right.
Way to go, Jake.
Go, Jake.
While you're there, you want to tuck that little chunk of rope there back underneath the porch that's been in my shot.
Yeah.
Oh, the chunk of line.
Have
Yeah.
better luck cutting it off with the coral cutters.
There, really.
Or use the knife.
Yeah, come on, go with the knife.
Yeah.
Oh, you gotta hit that one. Yeah.
While we're sitting here, can we try to get a shot of that anemone just so we get a little closer view of it before we fly away?
Yeah, sure thing. Awesome job. I can turn my head in because I don't think this will get us the shot we want. All right, Jeff, go ahead.
Something weird on the
Go ahead and push
this
on
side
and
of the anemone. Texture on that rock is very distinct. Another rock. It's part of the anemone, but there's pretty fresh tissue. The other part seems to be some sediment on it. Don't know why. I'm just making an observation.
Pretty. Thanks, Jess.
Yeah.
Okay, great.
Pull away, please.
Okay. So I guess we can put in that final ship move to get us over the end target. Do we have any indication if the feed has gotten any better? No.
Better. Okay. No better. Got a few loyal followers hanging on with us, though.
So for those watching at home, Jeff, could you put up the pack view on one of the quads? And Randy, do you mind zooming out just a tiny bit?
Sure thing.
So our initial dive plan was that we started towards the southern end of this volcanic feature, and we've been moving along kind of within 500 meter range towards the northern end, and we're getting close to that target now. So we're going to finish that final move, and then I think our plan is that we're going to move to the west, try to see what's over on the other side.
Cool.
Sounds good.
Thanks, Jeff and Rennie.
Are we going to potentially summit?
Well, we will try to go over that top, yes, of whatever is indicated in the multi-beam, whether or not it's an actual summit to be determined.
Yeah, we can, once we get the vehicles up there, sometimes we just scan around with the sonar until we don't see anything else, and then we know we're at least.
Plant a flag.
Yeah.
Yeah, I think this one goes up higher, so we won't get there, the lower one.
Is it possible to zoom in on that little swimmer in the middle of the screen?
Oh, yeah.
Ooh.
Sure. Cool eye. Yeah. Go ahead, Jeff. Very fast swimmer.
Oh,
Wait,
it's falling
Is it
apart.
south or
I don't know.
know.
colonial? Yeah, maybe a siphon 4. Siphon 4. Say siphon 4.
Oh, it was a
Well,
smushed up in the wash.
Either that or the lasers got them in hand.
Seems to be some trouble in the colonies. Disbandment.
Yeah.
All right. Come down a bit.
Yeah. I think we're going to keep going down for a bit there, Jake.
A nice view of the front of Herc and Argus cam. Almost like a Herc selfie.
It's not a really elegant way to descend down this without losing a lot of visual contact. Given the slope of it.
Okay, well, we're not going too far to this arbitrary target.
Yeah.
Looks like Argus is starting its northward move. Yep.
What are you off the deck? Oh, you're only 15 off. Yeah. You want to look down on me a little bit there, Jake?
Yep.
We just have a few more meters, 10 meters to go. See what's around the bend. We've kind of stayed within 100 meters or so of contour.
Mm-hmm.
Looks like there's another branched coral up ahead. Not branched coral, bamboo coral. I think my lack of sleep is catching up with me.
I'm going to do a quick gauge check.
Sure thing.
Jake, when you're checking gauges, what are you looking for?
So I'm checking basically the oil levels for a number of different compensators on the vehicle to see whether they're holding steady or changing.
Jess, can you pan to the left a little bit there?
Sure thing.
If they're changing, I expect like I don't know, an oil leak or something. We want to monitor that. But for the most part, they tend to hold steady. But it's just something we do periodically to make sure everything's okay on the vehicle.
And those gauges are directly on the vehicle, and he's actually just panning a camera around to look at them.
Yep.
Kind of eliminates one element of potential
error if you had a digital signal coming up to a screen to see. Kind of, we already have a bunch of cameras down there.
Good. Push on in a little bit there, Jeff, please.
Steve, what are we looking at here? You know?
I think we've got a dead sponge from similar to the ones we've seen
Squat lobster
so far, and then yeah, a bunch of persingid sea stars. Squat lobster, yep. And a couple of chrysogorgid corals. Our friend Romulogorgia and Chrysogorgia bottle brush morphology.
Mm-hmm. Thanks, Jess.
Yeah, sure thing.
And Jeff.
Oh, we hit an eerie moment of calm. You can see that Argus's sub-bottom just suddenly is just flatlining.
Quite a bit of shark activity by the
Yeah.
surface.
Oh, yeah.
Oh yeah, there are more sharks in the.
We all collectively have to remember to go take a peek at that.
Yeah.
Yeah.
Jeff, do you have the ability to show up the A-frame view?
It's the wire cam.
All right.
So our viewers at home
Yeah.
can see these swimmers up here at the surface of the ocean. Well, maybe they're a little camera shy now. Rennie, once the ship gets to the target, if we would like to go up to that little diamond shape that might indicate a summit feature, I guess a west-southwest move.
Yeah, can do. I'll just call that in right now.
Okay.
Do a little.
So definitely more sediment on this edge of the seamount. Might indicate that different current on this northern side than on the eastern side. But still some sponges. At least 3 of them in the frame of view right there.
That shiny thing.
If it'll push on there for a sec center screen. Can? No.
Oh yeah, some garbage.
Garbage.
Interesting. Looks pretty fresh.
All right. Go ahead, please.
Yeah, a lot more sediment here.
I don't know if you caught that. Just will be going generally the direction you're going. Go to 242.
242 range.
I think it'll just curve pretty shortly.
Jess, do you think, Rennie, do you think we have time to try to do a sediment short here? Even though we just called it in the ship's move?
Yeah.
I can cancel the ship move.
Yeah, if you maybe
Let's
pause
do
on
that,
it for
yeah.
a second. Figure, with this much sediment, I can't not track.
Yeah, let's see here. So again, Argus can.
Mm-hmm. Kind of behind us.
Yeah,
Right
probably
behind
right.
you,
Let's give it a try. I'm curious how thick this layer of sediment is.
Yeah, me too.
Yeah, probably actually down here.
The switchbacks.
You can
I
see,
know.
The tracks, huh?
See the tracks.
yeah, definitely some animal life moving around in the sediment here. Even some trails.
Arginers, you can cover, yeah.
Worm was, or yeah, something was going skiing. This little depression here is
Very
good.
interesting how the sponges are all on the sediment.
Yeah.
That's not the place you should be. And they're very unusual.
And then also no nodules here.
Today's opposite day. Sponges on
I'm
sediment.
going to come down a little in the delta there, Jake.
What's the plural of sponges? Like a group of sponges.
Oh, like the collective.
Yeah.
Yeah.
A linen
Mock.
closet.
A sink of sponge.
Yeah, under sink.
Value pack.
Valley pack of sponges.
Xenophyophore right there, though.
Oh,
Upper left.
for.
Oh, is that what that is?
Yeah,
Oh, wow.
a good sized one, too.
Is that at all a collection target?
No, I mean, not on this cruise.
Yeah.
But yeah, sometimes. I wonder what that little tuft is in the upper left-hand corner, though. It's curious.
Yeah.
You want to zoom in close up on the sediment before we take a core?
Sure. Yes, that sounds like a good idea.
Roger that. Go ahead there, Jeff.
That's great. Can we make sure we get some stills
Yep.
with that? Thank you.
All right.
Okay, let's go for it.
Awesome.
So these sediment samples will be used as another counterpart to the study of microbial life on these seamounts. Even though we can't really see it with our eyes, there
Follow me around a bubble there, Jake.
probably tens of thousands to millions of microbial cells in each teaspoon of sediment. And some of them might be eating the organic matter that's buried in the sediment. Some of them might be
I gotta switch cameras.
using chemical energy to fix new organic carbon. So we'll be
Other
collecting
salvo there, Jake.
some of the samples.
Oh.
To study that.
Yeah, let's do that one. There aren't any push cores that have been done, is that correct?
I believe that's correct.
All right, do you want to pop it halfway out there, Jake? Yeah, that's great. Great angle here. It's not the right angle.
Yellow tape or yeah, yellow tape.
Yeah, yellow tape.
Beth, can you elaborate a little more on the role and importance of the microbes? Do they free up nutrients that other organisms can't extract, for example?
That is one thing they could do. So microscopic life can actually be many different forms. So it can be from the domain bacteria, it could be from the domain archaea. It could also include microscopic fungi. And some of those organisms are known to have the capability to break down organic matter into more simple compounds that other organisms need. That's probably a primary role that they play.
Go ahead and push on there a flip it, please, Jeff. That's great.
They also cycle things like nitrogen and change the forms that nitrogen is in. And so depending on the organism, they might be more or less happy with those nitrogen transformations. If the sediments go anoxic, they also will release metals in dissolved form that can then migrate to other depths. And metals are definitely an important part because almost all enzymes have metals in their reactive centers. So all forms of life need metals in their diet.
Okay, that's about as far as she'll go, I think.
Okay, great. I'm going to hold my breath and keep my fingers crossed.
Oh, she's stuck in there now. Come on. Ooh. Full weather, please, Jeff. I don't know how much of this core is going to be left.
Yeah.
Yeah, it's all falling out.
Yep.
Need to rinse and try again?
I don't think we're going to have much more luck. But yeah, let's go try. Maybe slightly more to the right instead of dead center. So we're a little bit closer, swinging around.
Roger.
And maybe it's a little bit deeper over there. Who knows?
Sediment eruption there.
Yeah. So we're in a very nutrient poor part of the ocean. So there's not a lot of life above us, and so that means there's not a lot of dead cellular materials sinking down to the bottom or minerals from the ocean. So the sediment, even though it's relatively thin, is probably still quite old at the bottom.
What does it tell you that there's not nodules in this region right here?
Oh, I don't know what that tells me.
Push on in there a bit, please, Jeff.
It probably means, my guess is that here, even though it's still a relatively low sedimentation rate, the fact that there's so much more sediment on this side tells me that probably the sedimentation rate on this part of the seamount is probably high enough to preclude manganese nodule formation. That would be my guess.
That makes sense.
So something about the way the currents move around the seamount to allow sediments to accumulate here, not elsewhere.
This one is a bit stiffer, actually.
Yeah, okay. Well,
See, we're moving the vehicle already.
yeah, let's go ahead and try to extract it and see if we can retain anything.
All right, full wide there, Jeff.
Little plug.
You still want this?
If it'll make it around, let's just go ahead and try to shove it in there and see.
Roger that. Oh, no. Yeah, I don't like it does.
I don't think anything made it.
I don't think so either. Okay. Well, yeah, I guess just shake it out and put it back in the river, please.
Roger.
I tried.
Beth, we're 0 for 2 on these guys so far.
It's not deep enough.
Yeah. Well, we always knew it was going to be a challenge.
I wonder if you like, stamp it once, stamp it again. That's not helpful for science, but
Yeah.
Pretty cool try.
Divide your analysis by 2.
yeah, exactly.
So folks are already wondering about the seamounts we visited so far in this expedition have followed an age pattern.
I wish we had the answer for your question, but we don't yet. That's really a driving motivation for why we're out here is these ancient volcanoes are of a series that we think is kind of oblique and tangential to the Hawaiian island chain. And that may mean that they're older and they formed prior to the formation of the Hawaiian island chain. And so part of what we're trying to do is actually collect rocks from these seamounts so that they can be dated to tell us actually how old these seamounts are. So we don't yet know, for instance, if the seamounts to the southern end of this chain are younger or older than where we are now at the northern end of this set. So we'll have to wait a couple months or so, maybe a little longer, depending on facilities being backed up due to COVID to get these analyses done. But that's definitely a primary objective of this set of dives that we're doing. So great question. Stay tuned. So there's a variety of shore-based scientists that are part of that study, as well as our shipboard collaborator, Andrea Balbus, Dr. Andrea Balbus, who I believe is at Cal State Long Beach, and is part of that study.
Yeah, she'll be on the next watch here in about an hour.
Ready to continue?
Oh yeah, let's do it.
Yeah, I think we're done here. Thank you.
Which nav?
Rennie, are you calling in the southwest move?
Yes, the 242.
Yeah, great.
50 meters, bearing 242. Thank you.
Yeah, and it looks like we got Herc right to the end mark where Chris told us to end. So this is it, sediment. This is where Chris wanted me to come.
Steve, did you want to look at those two, the zoanth, or
if there's going to be time before we move, can we look at those two little tufts of things?
what is it? Xenophyre, or is that?
Yeah, the one on the right is xenophyre.
Go ahead, Jeff.
And
It's
tumbleweed.
a single-cell foraminiferan that makes a house out of agglutinated sediments.
So cool. Nice house.
It's a living thing. Nice. And then to the left, there was a tumbleweed. I think that might have been a ball of spicules from a sponge, or
Oh
that's what
yeah.
I suspect is former sponge location. Great, thanks.
Xenophyrophores can get very large, right?
Yeah, like softball size or larger.
That's interesting.
Yeah,
Yeah,
usually they're more common in areas where there's, or more abundant in areas where there's higher productivity, but I mean, you also find them in the sediment, but
Oh yeah.
perhaps not as large or as abundant, as dense in these oligotrophic waters.
Rennie, can you reiterate the bearing we're going to be hitting? 242, roger. I didn't do anything. Maybe it's just a bad contact in the back. Did you? Listen here, Jake. Yeah, center monitor. While we're at a standby, I'm just going to give myself a small car wash. So, ending my head really quickly. Sure thing. Watch the wall, Raj.
I was just about to ask you to do that, Jeff. Thank you.
Sorry?
I was talking to Jeff, bringing up the link on the camera.
Raj.
Whoa. That was really weird. Looks like Rennie's part of the computer.
Yeah.
Steve, have you ever been involved with collecting Xeno 504s?
Yeah, a couple of times. We did an expedition last year about this time in the California borderlands where we collected a bunch of Xeno 504s. I've also been involved with cruises off Costa Rica where we collected Xeno 504s, sometimes with bridge cores. Very effective. Just instead of picking it up, just core the whole thing.
So someone's wanting your professional opinion. I guess they dove on the Pescador events and collected Xeno 504s, wondering if you also noted that they produce a kind of oil and a bit of a funky smell, kind of like a truffle.
Well, I haven't noticed that. So I can't speak to that.
Are there gaps in the Pescador Basin? Could it just be the sediment itself that stinks?
Could be. You know, the Falcor just wrapped up a cruise there.
Yeah.
Recently, and I haven't really had a chance to follow what's going on in that particular cruise since we've been out here.
Stocked crinoid.
Yeah, that's some of those waters are toxic in that basin, so there's a number of things that don't smell very well when there's no oxygen around.
Yeah. Jess, could you pan left to see those? I don't know what that was.
Sorry, guys. I'm a little bit behind.
Okay, that's fine. Don't worry about it.
We're kind of troubleshooting a few issues with the computer as well. So
okay. Do your thing. We'll just watch the view.
Okay, sorry about that.
No worries.
I will put that on the list though to smell Xeno 504 next time I see one.
Okay. Thanks. I don't think you don't need an excuse.
Smell everything once.
For those who are watching the lab cam this afternoon, well, I don't know what that was. That was cool. We had our co-lead scientist, Chris Kelly, in the lab with us this afternoon, helping process our biological samples. And there was definitely a shot of him smelling a coral.
I'm always amazed, even dry animals like sponges, for example. They have just this rigid smell, sometimes years after they've been completely dried out. I don't know. I'm sure they're full of bacteria and all these nasty things, and they produce all these metabolites, but I just can't imagine why something that's been dry for sometimes years has such a strong odor still. I don't know, just an observation. I have a number of sponges from the lab and from work that I've done in the past. There's always been a question in my mind.
So the view that we're seeing here, both in Herc and Argus cams, is of a slope of this almost like a sheet flow, but it's covered in sediment. So again, definitely seems that there's more sedimentation on this end of the ancient volcano. We're now transiting towards the southwest, coming slightly up in height.
Yeah. This was an area that was just mapped earlier today.
Correct.
Higher resolution. And speaking of detailed seafloor maps, Randy, do you have time to take a question?
Sure.
Beyond just knowing more about the topology, maybe topography, what other benefits are there for the more detailed seafloor mapping?
Besides the topography, so we also collect information not only about the topography, bathymetry, but the intensity of the return. So we send acoustic pulse down, and when it returns, it comes back louder or softer. So that intensity can correlate, doesn't always have to, but can correlate with the substrate. So we expect a louder return off of a harder substrate, such as the volcanic rock here, and then a more sedimented, thickly sedimented substrate would come back kind of softer. It would absorb a bit of that sound. It wouldn't and deflect it. It wouldn't come back as loud. But that's a bit deceiving because you could have a thin layer of sediment over some really hard substrate, and you might go down expecting to see rock and not see any because there's just a thin layer of mud over it, sediment.
And I would add, there's the intent to, do high resolution mapping around these volcanoes, but also on our transit in between the volcanoes. And for instance, as we were coming out from Oahu towards this region and mapping, sometimes we're going over part of the terrain where the background maps, the satellite-based gravimetry maps, didn't indicate that there was anything, and we would see a small feature on the floor. So there's underwater hills and mounds and mountains that aren't readily apparent in the existing low-resolution maps. And, oh, well, here's a little spot of corals.
Yeah.
Finally. And so another kind of outcome benefit is getting a better idea of what the seafloor looks like, which is very important for navigation and avoidance. As we know, there are instances of underwater vehicles with people in them smashing into underwater features because they don't see that they're there. So that's another important reason that we need these maps.
Thank you. Both.
All right. Well, there was like 10 corals in a little 5 square meter area, and then they're on.
I can call in another move to head towards the summit. Is that good?
Yes, please. The presumed summit. Couple more corals. These all seem to be species that we've observed before. Relatively high density. Any interest in an eDNA sample?
Yeah, maybe in a couple of more seconds when we get into this and see how extensive it is.
Okay. Yeah, so Jess, I think we want to spend a little bit of time looking at this outcrop here.
Does it go up much more? Or is this just a local high point?
Looks like
more of a high point on this for this little mound, but it does continue to go up to our right side.
Let's keep going a little bit more.
Okay.
Yeah, I see. Now it's gonna level off.
Yeah. Looks pretty flat there in the Herc sonar.
Yeah.
What kind of coral is that in the distance? And that's upright. We don't have to go over there, but is there any way you can get
We're heading right in that
a
direction.
zoom on that fan in the back?
Yeah, we'll head right over there.
Yeah, we're heading in that direction.
A little out of scope right now. We can dream about it for a minute.
Yeah. Do you think it's a precious coral? Maybe. I can't tell from here.
What's the Latin name for precious coral?
The family name is Coralliidae.
Coralliidae.
While we're waiting to get closer to that fan, can we zoom in just a little bit to see some of these animals right in our front field of view?
Yeah, sorry. Going down. How about we take a look at the corals here while we're waiting?
Even though they're things I think we've seen before. It doesn't hurt to get some nice zoomed in images.
Yeah, just about every one of these Chrysogorgia colonies probably has at least one, probably two or three squat lobsters or shrimp associated with them that live inside the colony.
Anyone in particular you guys have your eye on?
I don't know. Steve, what would you like?
Directly below you, there was a far more bushier one that had branches. It wasn't a traditional bottle brush. It looked like it was a very, very bushy colony with a fork in it. Should be seeing it soon. I think it's the one.
Is it this one here?
Yeah, yeah, either one of those.
Okay.
They look like bottle brushes, but they have branches coming off in odd places.
So like this one? Or that one?
Yeah, both of them, either of them.
Yeah,
they're
the same thing. Maybe the one to the right. It's this one's kind of weird to look at because it's
looking at it top
yeah,
down.
you're at top down.
Sure. You're gonna give me a little bit of slack.
Yep.
Sorry, I'm a little far away now. You can come full wide there for a minute, please. Thanks.
I think if we want to get that precious coral, potential precious coral shot, we should.
Yeah, it seems like we need to go back up there.
Okay.
Yep. We're
Here, we can just do a
just
snap
waiting
here.
until you had some fish to get out there.
Yeah.
Yeah. Go ahead and do a really snappy one.
All right.
Thank
Looks
you.
good.
All right, let's look at the other one.